10 mile, 100 miles, and they zoom way out into the galaxy, and then they come all the way back down into them eating a picnic, into their hand, down in the atomic layer and things like that. I thought it was really cool. There's probably on video on YouTube somewhere. But sometimes I like to think about things like that to give you an idea of scale. So anyway, I stole that idea. So this is my powers of 10 introduction, So if you have like the Earth, 10 to the 7 meter, you zoom down in order of magnitude, you get something about the size of the country. And this is old. This is very old. I should update it. But it doesn't matter because it's super, I got it actually because it was a good graphic. But there's a huge amount of deaths each year from heart disease, and that hasn't changed. And calcium channel antagonists continue to be included in the top 200 drugs. So an important class of therapeutics. All right, now if you take the 10 to the 6th and you zoom down in pretty far, you zoom down to the scale of meters, something about 2 meters, maybe a tall person. These calcium channel antagonists have to do with treating ischemic heart disease, hypertension, and other types of indications. You zoom down in another order of magnitude, you get to the target organ, the heart. These can alter the heart and smooth muscle contractions, and we'll see how. All right, now we're going to take another big leap. If you imagine the heart was the size of the Earth, and then you zoom down into the size of a person there, you'll get something that is on the order of a protein, 10 to the minus 9th scale meters. And here's sort of a biological membrane. You see the phospholipids sort of shown in cartoon form. And the particular protein target of this class of drugs, the calcium channel antagonists or blockers, is called the L-type voltage-gated calcium ion channel. And we'll unpack that. Here I've shown it as a black box because when these drugs were first developed, it was a black box. No one had any idea of what this protein looked like. They had some idea, but certainly not in detail that would help for designing a drug. But this is what allows calcium to come through the membrane, this charged ion, but in a very regulated way to changes of voltage across the membrane. That's kind of strange. We'll unpack that too. It's a mechanism called excitation-contraction coupling in the heart. And it actually was a black box until maybe 3 years ago when someone finally determined a structure of this with some of these therapeutics bound. That was kind of cool. All right. If you zoom down even one more order of magnitude, about the scale of angstroms, there's a couple angstroms between each atom here. You get something the size that you're on the same scale as the therapeutics. First one shown here is verapamil. It was first introduced in the 60s. And we'll talk about how that works. Okay. So that sets the scale. What pathway are we interacting with? And that's this excitation-contraction coupling. And this is shown for heart muscles here. Again, this is my cartoon of the cell membrane, the outside and the inside, the cytoplasm here. And somehow you need to get calcium ions from the outside to the inside to stimulate these heart muscles to contract. And the protein that's important for that is the protein that we're going to be talking about, this L-type voltage-gated calcium ion channel. And we'll unpack in the next few slides how that works. But essentially, you'll get a voltage, a change in the charge balance across this membrane. One side will become more positive than the other side. That will trigger this channel to open, allow calcium ions to come into the cell. Also, that triggers the release of calcium from some intercellular stores that are stored up in the sarcoplasmic reticulum. Now what happens then is these calcium ions bind to this protein called troponin C. Now, normally, troponin C inhibits the interaction of actin and myosin that are important for muscles to contract. But when it binds to the calcium that come in, the calcium comes at a concentration enough to bind to troponin, causing it to fall off and allow actin and myosin to interact, and you get this muscle contraction. Now, calcium channel antagonists, or blockers, reduce or prevent the increase of this free cytosolic calcium by interfering with their transport through these channels. So you get effects in the heart and also in the smooth muscle that use some of these pathways as well. All right, let's focus now on this voltage-gated calcium ion channel, because that's the target of these therapeutics. There are a bunch of different types, but this one is called the L type. I won't go into the others. But when they were developed, a detailed structure wasn't really known, but there was a structure of a similar protein that allowed potassium ions to come through. Potassium ions here are shown in these green spheres. And that was known and shown here. So the idea was, well, probably based on sequence, because they're kind of similar, we think the calcium ion channel might have some structural similarities. And here's kind of a cutaway view. If you imagine here's sort of the plane of the membrane, this is kind of the outside, this inside. Both were thought to have kind of a similar protein fold, kind of wide entrances and exits. And they're thought to constrict in the middle, have a very narrow spot that gives the selectivity for what types of ions that can go in and through the channel. And also, they can sort of define what that ion is. And they also have some kind of switch to let them open or close. And that can happen in a couple different ways. And actually, we'll see it's not just open and closed. It's a little bit more complex than that. Okay. So what was known about the actual L-type voltage-gated calcium ion channel? And that's shown here, sort of a cartoon. Again, here's the cell membrane. Here's sort of the outside and the inside, the cytoplasm. You can see these are transmembrane spanning proteins. This channel actually is made up of 5 different polypeptide subunits, but the most important one that has all sort of the functioning bits of it is this alpha one, and it's the largest, has the machinery that makes it work. And this is kind of interesting. It actually has 4 repeats of what was predicted to be these 6 transmembrane spanning domains. So repeat 1, 2, 3, 4, all attached together. If we zoom in, there's something kind of funny about those. Maybe I should do this for powers of 10. You can just keep zooming in. Actually, now it's probably Google Earth. I should probably use Google Earth. That's the more contemporary reference. All right, focus, 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 focus. All right, a little blurry. So here we have the 6 spanning, what was predicted to be the transmembrane spanning helices, looped by these loops in either side. And what's unusual about these is, in the 4th one here, there's a lot of residues that have these positive charges. Now that's really weird, right? Because you're predicted to be these transmembrane spanning the helices. You're in with all the non-polar acyl chains, the phospholipids, but you got these positive charges in there. That seems really energetically unfavorable. It's weird. And we'll see later, this positive charges are really essential for this protein to be able to sense the change in voltage. So this is actually the key to the voltage sensing of this protein. The other thing that's weird is in between these loops, these predicted helical sequences, 5 and 6, you can see that the protein kind of loops into the membrane, comes back out, and then goes through 6 again. And they determine that by a variety of experiments that aren't worth talking about right now. But the idea is that maybe these loops, which are called P loops, all come together. The 4 different repeats of these all come together, and those might provide the specificity of the protein. So just a P loops specificity. And these are repeated, in each of the domains. Yes, sorry. I just write P loops specificity. Yes? For which type of ion comes through. We'll unpack it. Okay, now the protein isn't typically splayed out like this, all in a line. So the idea is most of these transmembrane helical proteins actually pack together in bundles. So here's sort of the predicted packing of what this looks like. The alpha-1 subunit, which will focus on the middle here, each of these have these 6 transmembrane domains. It's thought that each of those pack together in a little bundle, and then you get 4 of these bundles that come together, and to make a central pore. So it's thought that there's a little space in the middle is where the calcium ions come through. So the alpha subunit packed together in 4 bundles. The central pore is lined by residues from each of those 4 bundles. And those P loops, which might provide the selectivity for which ion is let through, all sort of face the middle of that channel, providing that selectivity. And if you sort of zoom in on that, there's one model here of how this calcium selectivity works. Okay. So the P loops sort of dip in and out of the membrane but don't cross. And this particular protein, they contain these highly conserved acidic residues, so glutamate or aspartate, and those carboxylates on the side chains. And they're thought to make up this metal binding motif that determines which ions bind and which come out. Now, this was studied in a couple different states, which is kind of interesting. If you have sort of a state that's not physiological, where you have super low calcium ion concentrations, usually you don't get to this. This is a lab artifact, I guess. The channel can actually pass sodium ions through. It doesn't go through, so it's not super selective. But when you have sort of physiological ranges of calcium there, one calcium ion binds to the residues in this P loop, and it's high affinity, so it binds really, really tightly. And by binding there, all these other ions, like sodium, that might otherwise pass through, boink, can't go through the channel. So that's neat for excluding other ions, but what we really want to do is transport the calcium ions. So what difference does it make if you hold one tightly? You have to find some way to move it along. So what they found is when you can increase calcium ion concentrations, the idea is that a second calcium ion binds, and then this first site, which was originally high affinity, now gets converted into a low affinity site, which then can get released to the intercellular side of the cell. Now that's just one model for how these can, achieve selectivity. There's other competing models. I'm not going to go to it in detail, but I think it helps to have sort of some idea in your head and how these channels can distinguish one thing from another and regulate ions to come through. Now, the other thing that's weird about these is the idea that they can allow ions to flow through depending on change in voltage. I mean, usually proteins, I think of, a ligand binds to it, you get a conformational change and it opens or it closes. I mean, that's kind of easy to understand. They're all sort of physical interactions. Something binds, it changes the conformation, allows an ion to come through. But here, it senses whether the outside of the membrane becomes more positive or more negative than it used to be, and somehow the protein changes the conformation in relation to that change in charge across that membrane, the change in voltage. And this is a way to tie the conduction of a nerve impulse to these conformational changes that let ions flow in and out. It's really kind of cool. Again, the structure of this L-type calcium ion channel, the target of the calcium channel antagonists, wasn't known, but a similar one was this voltage-gated potassium ion channel, same kind of voltage gated. And that gave them an idea for how this works. And it was a little bit of, I don't think we need to get into it, but before it was predicted that those positive charges were in this transmembrane helix that moves up and down. And there were all different models. Does it screw? Does it just go up and down as a piston? Well, actually, when they got the structure of it, they said, oh, it doesn't do any of these. It doesn't look like a helix at all. So they had to sort of change their structural models. And it turns out they make these paddle-like structures on the outside of the protein. Here it is in two states, open and closed. And you have these positive charges. Now, that's kind of unfavorable. They're just buried there in that plane of the membrane. So what happens is, when the outside of the cell gets a little bit more negative than it used to, these positive charges are drawn upward by that electrostatic attraction. That leads to this conformational change, which can then open the channel, and then the calcium ions can come through. So these S4 segments that have the positive charge really are the voltage sensor, allows these conformational changes to result as you change the charge across the membrane.
I have a question.
Yes?
You said when the outside of the cell becomes too negative, that's when the positive charge is?
Yeah, not negative overall, but more negative. I put delta. More negative than it used to be here, because it's just sort of relative to each state. Negative, positive attract. So that's why you get the movement of that cross. Now, I talk about open and close. It's really not that straightforward. There's at least 3 conformations in the cycle of this. And I think not this lecture, but one down will unpack the different states to assign them to sort of structural aspects of how this happens. But actually, you have a resting state of the enzyme where there's no, of the channel, where there's no calcium ion flow. You get this voltage sensor, and then they open up, allow calcium ion flow, but then rapidly they get converted to a third structure that is called inactivated. It's still not reset to the resting conformation, but it doesn't allow calcium to come in between. There's like a third process that happens to happen. So it's not just quite the simple open and close. It sort of gates between all these different conformations, the resting, the open, and the inactivated. And actually that's important that it has to go through these conformational cycles, because that's how part of these antagonists work, is they interfere with this interconversion. Okay. Oh, good segue. All right, so how do these things actually work? Calcium channel blockers. There's other aliases for these antagonists, calcium channel blockers, antagonists. You'll see them called sometimes slow channel blockers or calcium entry blockers. That's kind of why I like this, because it's very descriptive. Anyway, the antagonists interact at specific sites on this alpha-1 subunit and block or inhibit the flow of calcium ions through the channel. Now, each class of the calcium channel antagonists that we'll look at actually have a separate binding site on that huge alpha-1 subunit. But because this whole protein undergoes these conformations between the resting, the open, and the inactivated state, a lot of these binding sites, binding at one site can impact the function at another site, because you're kind of blocking the motion of that protein. So they're allosterically linked. Sometimes it means they're close together. Sometimes it just means that they're coupled through these protein motions. Okay, so we'll look at 4 classes of calcium channel antagonists this morning. We'll look at the 1,4-dihydropyridines. You can see this used to be a pyridine ring, but when you're 1,4-dihydro, you just have these 2 double bonds. We'll look at the benzothiazepines, phenylalkylamines, and the diarylamminopropylamines. And this class, we'll look at a few, but most of these we'll just look at 1 or 2 examples. And we'll use this as a roadmap to show where we are. So first, let's look at this class, the 1,4-dihydropyridines. All right, so here's a whole bunch of them. Nifedipine is a nice one to keep in your head as sort of an example of the class. But if you look at all these structures, you'll see some things are kept in common. Remember we used to figure out the pharmacophore by superimposition? Well, here you can kind of do it in your head. What are the structural features that are the same to all these? All of these have this 1,4-dihydropyridine core in them. All of them seem to have these esters at the 3 and 5 position. Some of them are symmetric, but some of them are not symmetric. Oh, I should update this. Me equals a methyl. Some of these, see ET, that just equals ethyl. I don't want to be confusion. This symmetricality is kind of an accident for how these were synthesized originally. Some of these are now asymmetrical, so they don't necessarily have to be symmetrical. You just have to have esters in these positions. You'll see an alkyl group at these positions. And what's really interesting is sort of para to the nitrogen here. You'll have this phenyl group, and the phenyl group always has a substitution at this ortho or meta position. And we'll actually see that that's actually important for how these things, their conformation and how they bind. Okay, now let's focus in on nifedipine as sort of an example for the whole class. If you look at the structure flat, to me, come on, there we go. It kind of looks, flat. But if you, if you imagine the structure, that's actually tetrahedral carbon there. So if you look at how it looks in 3D, here's from the side, this phenyl group kind of sticks up tall, sort of almost perpendicular from the plane of this ring. And that's important for their pharmacophore. And the rotation that you get around this bond, right here, is actually restricted because you have this substituent at the ortho or meta position. And that's actually important for the activity because it restricts whether this phenyl group can spin around or if it can just point one way, back in this direction. And I'll pull up Chimera because I think that gives you a little easier, can view that a little easier. All right, so here's the nifedipine. You can see here's the dihydropyridine ring. Here are your two esters symmetrical on either side. And then you can see that the phenyl ring is almost perpendicular. Burp, burp. And then here's the nitrosubstituent that sticks back. And you'll see it kind of sticks in between these two groups. And it actually holds this position in that perpendicular orientation so it doesn't just spin around like that. And I can probably show that here. Atoms, bonds, sphere. Okay. You see how that nitro group is just kind of snuggled in between those two ester groups there? Not a lot of room. So if you tried to rotate that, I don't want an update. Try to rotate that like this. Oh, I'd run into it. I can't go over there. I can't go over there. So it kind of sticks it right in between these two, which is kind of cool. There is actually one other orientation that works. You can see it kind of crashes into these groups on this side. But if you put it right over the ring, it actually works as well. But it ends up having the phenyl group in the same orientation, that same perpendicular orientation. So either one is favorable, but you still get this orientation. So that's sort of why you need this substituent on that phenyl group to hold this in the right conformation for interacting with the target. Okay. And we'll see that's important because in metabolism, you'll change the geometry of this. It'll turn into a flat structure, and then it doesn't bind to the receptor anymore. But we'll unpack that. Oh, yeah. Okay, so I made a big deal about we don't have no idea what this, when they made this, we don't know what the structure looks like. We don't know where the binding sites are. So how do you figure that out? There are some tools that were used a lot in this field, which I think are kind of interesting. And these aren't drugs. These are sort of methods for telling where drugs bind. And one of these methods is called using photoaffinity probes. And this is just sort of a general description. You'll have a group such as this. It's called a phenylazide. And when you shine light on this, certain kind of light, it makes a really reactive species. I'm not going to ask you to know the chemistry, but I do kind of want you to know the technique. This makes something super reactive, and what it will do is will react covalently with residues in the protein to give a new covalent link between the protein and the probe. So these are kind of like tools for finding out where drugs bind. So the way these are used is you take your drug and you put one of these photoaffinity probes on it. You mix it with your target, here membranes that have this calcium ion channel in it. You treat them with light to make that react with species, and then it makes a covalent bond between your drug and your target, and you can figure out which amino acid that gets attached to, then map it onto the protein sequence and sort of figure out where your drugs bind. You can do this without knowing the structure of the protein. This was done with the dihydropyridines. We talked about making these asymmetric substituted esters. Here they've just put a photoaffinity probe out on this one arm. And they find that they modify a bunch of residues in this P loop and this other S6 transmembrane domain. So they think, oh, it must bind near S6 and maybe sometimes near this P loop close to the selectivity filter that lines that calcium ion pore. Now, one caveat with these are that you're adding this big group to the drug, so it might not necessarily bind where it normally does because you might perturb the system. And that's just a drawback of the method. Okay, so then in 2016, a bacterial protein that was found that was a voltage-gated calcium ion channel. This is used as a model for how the human protein actually looks. So this would be that alpha-1 subunit made up of these 4 repeats of those transmembrane domain spanning regions, and that seems to be true. And the recent structure shows that this binds right here, which is where the nifedipine binds. And it's actually binding close to the S6 helix on this P loop, but it's kind of interesting because it's on the periphery of the protein. So here's sort of a side view in the plane of the membrane. Here's kind of a top-down view. Here's where the calcium ion channel is in the middle. You can see it kind of binds here where in the plane of the membrane, where the lipids normally pack. So it makes a lot of hydrophobic interactions, and it's a lot of steric constraints that fit the exact shape of that pocket. Let me pull this up. Okay. I think it's this one. Okay, so let's zoom out first. So here's the channel, which is kind of cool. Oops. You can see the ions right in the middle. So this is kind of your top-down view. If you look on the side, and actually we talk about these 4 repeats. 1, 2, 3, 4 of these bundles that come together. And if you kind of look on the side, where the lipids would pack, that's where this nifedipine, or excuse me, this 1,4-dihydropyridine is bound. And you can see that you do have that phenyl group kind of sticking perpendicular to where that the reduced pyridine is, and that's because it has to fit in sort of this channel there. So that's why it's sort of that holding that conformation is important, because it has to sort of sterically fit right into this binding site. And by binding there, you can inhibit the conformational changes of this protein needed to transmit these calcium ions through. So the 1,4-dihydropyridines this is the other take-home for this. It's kind of interesting. They inhibit calcium ion flow, but look, they don't bind right where the calcium ions do. They go over here. They don't block it up like a plug. They sit over here on the side, and they prevent the protein from undergoing its conformations to let those calcium ions flow through in the middle. Okay. So that's what this class looks like. That's where they bind. That's their target. How do they get metabolized? Well, one of the main metabolisms is this dihydropyridine gets oxidized back to the pyridine. Now here you've got this sp3, it's sort of tetrahedral. The phenyl group sticks up perpendicular. Here you go back to sp2, so it's planar, and kind of it moves down from this position to something that's in plane with the pyridine ring. And once you do this, it's now inactive. And you can see why, because it wouldn't be the right shape to fit in that pocket anymore that we could visualize. After that, you can get hydrolysis of this ester to make the carboxylate. Sometimes you can get oxidation at one of these positions. And it's kind of interesting, you can even cyclize to make the lactone. But all of these are inactive after you lose that important conformation of that phenyl ring that's held in place by that substituent. Let's see. The enzyme, the cytochrome P450, is 3A4, and that's one of the enzyme that can get inhibited by components in grapefruit juice. So sometimes that's an interaction with this class of drug. Yes?
Just to clarify, on like the last slide, the last thing we were talking about, so the antagonist is, when it binds, it's stopping the channel from like moving that paddle up, which is not allowed, so then it stops the calcium from going through?
I'm not sure if it stops from moving the paddle or moving the inactivation gate or what the conformational is, but the idea is it's not sitting like a plug in the middle of the channel. It's somehow blocking the function of the protein allosterically. I don't think we have to get that much in the detail. Okay, so that's the dihydropyridines. Let's go next to the benzothiazepines. This will just look at one briefly, and that's diltiazem. It's kind of has this interesting 7-membered ring, which is kind of unusual. Here's sort of what's known about the pharmacophore. You need this basic group on here, pK about 8, on this nitrogen appended to the lactam. It's aryl and ether groups and the basic are kind of on the same face, so the idea is the 7-membered ring might just be a scaffold to hold all these things on the same face and maybe isn't that important for binding to the protein. If I look at this, if I think about what's probably the most metabolically susceptible group, I would look on here and I'd say, oh, we've got an ester here. That probably gets hydrolyzed. And it does, and the product is still a little bit active. 20 to 50% as active as the product, but that's one of the first sites of metabolism. All right, that's just briefly the benzothiazepines. Now let's go to these phenylalkylamines. And for this, I'll show the verapamil, which we used in that Powers of 10 introduction. Now might be a good time to take a little mental break and do a think, share, pair. So take a look at this. Here's I drew verapamil, gave you some information about it. Why don't you take a minute and see if you can answer the questions to yourself, and you can talk about it with the person next to you, and then we'll come back in class and discuss this. Okay. All right, it has a pK of almost 9. Which atom? This one? No. That one? Yes. That one? No. This is the one. Tertiary amine, so an amine that has 3 substituents. So that's going to be in equilibrium with protonated form. At pH 7, which form will predominate? This one? This one? All right, you guys are good. So that tells you overall charge +1 predominantly. If charge, where is it located? Right there in the middle. How many chiral centers are there? How many say 1? How many say 2? How many say 3? All right, 1 or 2. All right. Here's 1 because it's marked, right? Where's the other one? Someone that raised their hand for 2? What's that? Oh, the tertiary amine. Okay, I'm going to stick with 1. Tertiary amine is a little bit too far down in the weeds. It actually can equilibrate at the room temperature. If you slow it down, it will freeze into one chiral position, and you can see them separate on the NMR. It's really kind of cool. So yes, you're awesome, extra credit, but we're going to stick with 1. I could do how many chiral carbons are there? All right. Now, is it S or R? How many people say S? How many people say R? Oh, good. We have a debate. I love it. All right, let's do it. All right. So each of these have 1 carbon attached, so that doesn't help you, right? And then here is the carbon, and then it's attached to 3 other carbons. Here it's a carbon attached to 2 other carbons, so there's a proton there, and that would be a point of first difference. So this one is less than this one. This is a carbon attached to 3 nitrogens, so that's definitely less than this one. And this one has 2 protons. So if you go next, it'll have a carbon attached to a carbon versus a carbon attached to another proton, so this is the lowest. So I'm going to put 4, lowest priority. This one, 2, and then 1 will have nitrogen attached to the 1. Now, you probably would have known I'm lazy, so I put the lowest priority in the back usually. So anyway, it's not a good way to tell, but I put the lowest in the back. So, and then if you come around, you can see you're coming here counterclockwise or to the left, which is going to be sinister or S. And the S enantiomer is more potent, but the compound is actually marketed as a racemic mixture. Now, does that assignment make sense to people? Yes? Okay. All right. Now, what's kind of interesting about this, if we look at the metabolism, that stereoselectivity plays into metabolism. And also, we found that how it's dosed plays into metabolism. An oral dose is less potent than IV dose. The N-methylation and N-demethylation, sort of pulling off that, and O-demethylation, where you take off one of these. Where's that? I guess I don't show these. But the N-demethylation and O-demethylation are not stereoselective. So it's marketed as racemic mixture. Both of these enantiomers get metabolized the same through those routes. But there is an N-oxygenation reaction here, where that nitrogen gets oxidized. And then that can lead to a cleavage event here. If you pull off one of these protons, you can break into these 2 pieces which are inactive. Now that is stereoselective. So that N-oxygenation, it prefers the S. And that occurs during this first pass hepatic metabolism. It leads to less availability of that active S form in the oral preparations due to that metabolism. And that sort of underlines these differences.
Second structure there still active, or is that inactive now because of the...
This is a very transient species. I should put it like this. Because once you activate the center, you've got this positive charge. It's a really good leaving group. You don't need an enzyme to do this. It's just non-enzymatically can deprotonate and cleave. So it's not a long-lived enough species to worry about. Active, transient. Okay, so verapamil, it also same target, L-type calcium ion voltage-gated channel. Where does it bind? Well, let's use that photoaffinity probe technique. So I had a question during the discussion. The photoaffinity probe is just a method for people to use in the lab to sort of figure out where these bind, using purified proteins. So you have a purified protein, you make this artificial modified drug, you mix them together, and you can figure out where the binding site on that protein is. And that should reflect in vivo where that drug actually binds to that protein. But it's sort of used as a model for that so you can figure out where they bind. So here, verapamil, this is kind of clever. They took one of these phenyl rings and substituted it with the aryl azide. So here it's even more isosteric than the other one was. And this is used as the photoaffinity label. And they find that verapamil binds again near this S6 transmembrane domain. So they bind somewhere near the 1,4-dihydropyridines in repeat 3, but they seem to be different sites. And this is much more visible when they look at the crystal structure of this, which just came out just a few years ago. And this is actually, even though that method said that there were similar areas, it's actually quite a distinct site. They must be linked through these conformational changes. The photoaffinity label results are confirmed because it binds between subunit 3 and 4. But now, you can see here's the calcium ion channel. It actually binds right in the middle of this, and almost like a cork, because it binds right to prevent those calcium ions from coming through. It's sort of a more direct inhibition that's easier to see. Although most of these sites are linked by conformational changes, verapamil more directly blocks this flow of ions because it just gets right in the way of all these calcium ions coming through. So if we look at this, I can pull up the structure. So if we zoom out, I guess we can see there's kind of the top of the pore of the protein, the 4 repeats that come around it. If you go through a side view, you can see there's kind of a very constricted part of the channel that opens into this cavity internal to the protein. You can see all the ions kind of lined up coming through there. And then you see the tertiary amine of verapamil just being the next one in line. And there are some hydrogen bonds which hold this in, some hydrophobic interactions. What's kind of interesting is you think these calcium ions are positively charged, and you can see maybe why you need a positively charged group there in the drug. It almost acts like the next ion in the chain, right? Just comes right down there. Anyway, so it binds a very different, distinct site on the same target, distinct binding site. All right, so we did these classes. Now we'll finish up with the diaryl aminopropylamines. Unlike others, Bepredil, which is in this class, it also can block other channels, these fast sodium ion channels, receptor-operated calcium ion channels, not just the voltage-gated ones that it's sort of shown in this class. And that's kind of interesting if you are curious about how ligands bind. But it's not so interesting in terms of drugs because that means it can lead to a lot of different side effects. And this tends to have more side effects than some of the other ones because of this broad selectivity. All right, now just sort of I mentioned this, but I'll just bring it out again. We can see that the 1,4-dihydropyridines, the benzothiazepines, the phenylalkylamine binding sites are all sort of different on the same receptor, although some can antagonize or agonize the binding of others. If you bind at the dihydropyridine site, it actually enhances binding of benzothiazepines, but it inhibits the binding of phenylalkylamines. So you can have these allosteric to, each of these can inhibit calcium ions coming through, shown by that negative sign, but sometimes the binding of one might enhance the binding of another drug in that class or prevent the binding of a drug in a different class. So these are all distinct sites, but they're linked in sort of a complex way allosterically. Let's see, what about this? So we have these three states of the channel: resting, open, and inactivated. Now, verapamil and diltiazem, they're charged, and they don't bind to the channel in the resting state. So these are called, they only bind after it's open, which is kind of interesting. I think of that like, you have these calcium ions that have to get to binding sites. I don't know if this is how it works, but if it's open and you bind these positively charged things, that means these drugs can find their binding sites inside the protein. So if the channel is in use, opening and closing and inactivating, it'll be easier for this drug to find that interior binding site on the protein, one in that channel buried in it, and inhibit. So these are called use-dependent drugs because the channels that are more active are inhibited to a greater extent. But nifedipine, it's called the use-independent. And you remember that one's kind of neutral, sort of sticks on the outside of the protein, diffuses through that lipid bilayer and finds that sort of nook and cranny on the outside, this hydrophobic pathway. And that happens whether the channel is opening or closing. So it's use-independent. And this will get unpacked by other faculty as they come down to show why you might choose a use-dependent or a use-independent therapeutic to help different arrhythmias. Okay. Just for the last minute, sometimes I like to say what's coming down the pipeline. The current calcium channel blockers act on the cardiac L-type calcium ion channel, but there are a lot of other channels, N, P, Q, and R, that have locations in the brain. And specific inhibitors of those type have a lot of applications in causing pain, relieving pain, depression, controlling seizures. Now, there haven't been any drugs for these subtypes that have made it through the approval process yet, but some are coming. And there are some natural products which are kind of interesting that bind these N-type calcium ion channels, and these are called these omega conotoxins. This is kind of fun. I thought I'd put it on here. They're sort of these small peptides, and they kind of look like a cork, and they actually work just like a cork. They plug up right in the middle of that calcium ion channel, very unlike the dihydropyridines, which are these allosteric ones. These just bind right at the site. And where they're found is in these snails, and they actually use it to eat these fish for lunch. So snail's very slow, right? Fish is very fast, but it can harpoon this fish and inject the conotoxin and then basically screw up all their heart muscles, so they have to sort of slow down, and then they just engulf them and eat them. And there's a cool, it's not gory, but it's, maybe I should warn you, but anyway. So here's the snail on the left-hand side of the screen, and you have a little harpoon to the fish. So he'll harpoon the fish, and then the fish will kind of slow down as the conotoxin, and then he just sort of engulfs it and eats it. And I guess the snail isn't susceptible to the same sort of inhibition of its calcium ion channels. But that's kind of a cool, maybe very practical application of the conotoxins. All right, so let's finish up. DocHam, here we go. So again, here's my take-home points, the summary. I'll look at this when I write test questions. What do the voltage calcium ion channels do? How do they work? How do they get regulated? There's 4 major classes of these calcium channel antagonists. That's just my preference. You can use blockers. We looked at their structures, their in 3D, how it's important sometimes, like with the dihydropyridines. Their pharmacophores, their stereochemistry PKs, how they get metabolized, and what type of interactions they might have. And then we just briefly talked about this experimental technique using photoaffinity labels as sort of a general way to find drug binding sites on targets in the absence of structural information. Okay, any questions? Yes?
Do we have to know how to do the arrow pushing on exams?
No, this exam you don't have to know how to do the arrow pushing for photoaffinity labels. I might ask you something related to that N-oxidation where the metabolism cleaves that compound, but I'm not going to have you draw the arrows. It'll just be we're doing multiple choice format, so it's just going to be understanding how that process works. Anything else? All right, thanks everyone.
Good morning, everybody. Happy Monday. All righty. So I believe this is where we left off on Friday. Thursday? Friday? Sorry, I'm in a time warp. So we're in the middle of the beta blocker class of drugs, and I had talked a little bit about their different properties. I had discussed that they have different ISA, which is agonist, antagonist activity. So they function more as like an agonist when the patient is at rest and an antagonist when they're exercising, okay? The other part, one of the other differences I wanted to go through is the different generations of beta blockers, and I talked a little bit about the first generation of beta blockers that came out and discussed a little bit about if the patient's on a beta blocker and they come in and say their blood pressure is lowered and you want to increase it. The other things that you kind of need to be aware of with beta blockers is if you have patients that are at risk for developing hypoglycemia. Has anybody here experienced an episode of hypoglycemia? What does it feel like?
So your head starts feeling like a little bit more, you get lightheaded.
Okay, any other?
I don't know if this happens for a lot of people, but like the room starts to get dark.
Oh, okay.
Or you start trembling.
Uh-huh.
A lot.
Okay, right. You get trembling, you sweat, you get like a sense of urgency, like you need to eat something like really, really fast. So if you think about that, a lot of those are sort of symptoms that are associated with sympathetic nervous drive, right? So if you're on a beta blocker, you get hypoglycemic, you're less likely to feel those symptoms, right? And hypoglycemia is a serious thing you should do something about fairly quickly. So you don't really want to block those symptoms for patients who would be at risk for developing them. The other patients at risk where you would be blocking symptoms were patients who have pheochromocytoma, which is a pretty rare condition. Clonidine withdrawal, I'll talk about that a little bit later, and cocaine use, which cocaine is going to induce a large sympathetic stimulus, okay? Okay, so then came the second generation beta blockers, and what I want you to remember about the second generation beta blockers is they are more beta 1 selective. Okay, so these have good properties in terms of what we want to use them for elevated blood pressure. So they were an improvement, but both with the first and second generation beta blockers, you do see some increase, can see some increase in lipids, a decrease in heart rate, which we'll talk about in a little bit, and an increase in new onset diabetes mellitus. And I put atenolol here because it's a second generation beta blocker, and it tends to be one where you see a lot of studies coming out that are used for comparison. The other one that's used pretty often is metoprolol. And then lastly, we have the third generation beta blockers. So what I want you to remember about the third generation beta blockers, if you think about beta 1 blockers, they would mostly be working on the heart, okay? The added benefit of the third generation beta blockers is they have beta 1 selectivity, but they also have properties that cause vasodilation, okay? Now, how they do that might differ based on the third generation drug. So if you take carvedilol or labetalol, they're racemic mixtures, and some of those, block the beta 1 receptor. The others block the alpha receptor, which would cause a reduction in peripheral resistance, okay? Now, then we have nebivolol, which is beta 1 blockade, again, but it produces NO, and we'll talk about NO a lot when we get to the heart failure section. But remember, that's how you get local vasodilation. So that's another property that would allow you to reduce beta 1 activity as well as having an added benefit of peripheral dilation. Okay, tell me a little bit about, this is something I want you to remember for almost all of these drugs for hypertension. What do we know about adrenergic receptors when you block them long term? I'm sorry?
Body compensates.
How does it compensate? Okay, you're right. The body compensates, and we'll talk about that in a minute. But what does the cell do?
So does it increase like the receptors?
Yes. Okay, so the cell is going to upregulate the receptors, right? Because that's how it's sensing that it's not getting enough stimulation. So you get upregulation of the adrenal receptors. So when you have a patient who's on these drugs, you need to warn them not to suddenly stop their medication. Because if the beta blockade goes away, they have that many more receptors on the cell, which makes them particularly sensitive to having elevated blood pressure once the drug is out of their system. Okay? Yes?
And that's just for the third generation because
No.
the third generation, that's all of them?
It's for all of the adrenergic
Okay, so
receptor
not just the alpha
blockers.
receptors, all
Yeah.
okay.
Yeah. And I would actually tell the patient globally not to stop their blood pressure medication, right? Because the patient would be particularly sensitive to, but they really need to understand the importance of keeping their blood pressure down, which means like if they didn't get their refill over the weekend and they need a couple pills to get through, you help them with that. Yeah.
So would they have to be tapered off some of
Yes,
these?
you have
Okay.
to taper off. You have to account for that. Any other questions? Okay. So again, when we talk about beta blockers, the possibility that you would decrease the heart rate, these third generation beta blockers, less likely to decrease heart rate. All right, so as a group of drugs, things you'll probably talk about a little bit more later, but if you look at differences in how different patients respond, I had talked to you about the ACE inhibitors and how they were attractive for patients who have renal insufficiency or diabetes. Beta blockers, African American patients tend to not respond to beta blockers as well as other patients. So maybe this is not one of the better drugs for you to use in an African American patient, but there's other alternatives. The other thing you have to think about is while we decrease the blood pressure, our body relies on these adrenergic receptors for other effects, right? So say we have a patient who has heart disease, we're trying to ask them to get out and exercise more, maybe lose weight. One thing you have to keep in mind here is if a patient's on a beta blocker, it's more difficult for them to reach a higher heart rate. Okay? So patients might complain that, they can't really exercise as much as they wanted to. Not like they're going to be out there running marathons or anything, but something you want to keep in mind, that it would limit their ability to increase their heart rate during exercise. They might cause fatigue or depression. And then the metabolic abnormalities that I've already discussed. And then when you compare it to some of the other drugs we'll talk about, one of their benefits is they don't really cause retention in sodium and water as much as some of these other drugs that we'll be talking about next. Okay, so water, sodium retention increases blood volume, which then is going to rebound into increasing blood pressure again. So it would limit the effectiveness of the drug. So we like this property of the beta blockers for this reason. And then sometimes you can see patients with hyperkalemia. Okay? Again, this is an FYI table. It's kind of summarizing these different properties that I've talked about and why you would pick one drug over the other drugs. This timolol here at the bottom, it is a topical beta blocker. Okay? So I've never seen it anything otherwise. Okay. So that's the beta blockers. Any questions on those? Okay. So a little reminder here. Oh, and I brought my pointer today. Hold on. I'm going to get used to this classroom just as soon as I'm done teaching in it. Okay. So a little reminder here on the alpha receptors. Okay, you guys might remember all this. All right, so we have two different alpha receptors, the alpha 1 and the alpha 2 receptor. These are peripheral, these are central. And then whether we want to develop drugs for them, if we want the alpha 1 agonists are what we use for the treatment of nasal congestion, okay, because these receptors are in different places, okay. Alpha 1 blockers are what we use for hypertension because they're located, they regulate blood pressure in the periphery. But we also use the alpha 2 agonists, so don't get these confused. One is an antagonist, one is an agonist. And this is treatment for hypertension as well. Okay? All right. So as you probably recall, here we have the sympathetic nervous system, and it releases the norepinephrine. And what happens is when there's enough norepinephrine in this junction, then the alpha 2 receptor is stimulated by norepinephrine and then turns down the release of it. Okay? And then in the smooth muscle, we have the alpha 1 receptor that, when it's stimulated, is going to increase contraction of the smooth muscle cell in the vessels. Okay? So if we use an alpha 1 blocker here, then we ought to decrease smooth muscle contraction in the periphery. Okay? And we do have alpha 1 antagonists, so what they're going to do is reduce arteriolar resistance and increase venous capacitance. Do you remember all those terms from last year? So that would drop blood pressure. Okay? And then when you increase venous capacitance, what is that going to do to the heart? Huh? There's a term we use for how much blood goes back to the heart. Preload. Okay, so it's going to reduce preload. Okay. So there's 3 of these alpha 1 antagonists. There's prazosin, terazosin, and doxazosin, these are the common trade names for these. Minipress, or the one that's been out the longest, has the shortest half-life. I prefer that you just learn Cardura, okay, for the test. Okay, so if you think about this and you're going through what could be the possible compensatory effects if you were to give an alpha 1 antagonist to a patient, what do you think might happen when you increase or increase the vasodilatory effects of both the arterioles and the veins? What is the heart going to sense, and how does it sense it? What monitors the pressure of the blood going through the vessels? Pressure, keyword. Baroreceptors, exactly. So when you have a decrease in peripheral resistance, one of the problems that'll happen with using these drugs is you get the baroreceptors becoming stimulated, which are then going to do what? Send the message to the vasomotor system to increase sympathetic nervous drive. Okay, this is why it's a second-line drug, because it's not very effective when it's used alone. Okay. So when those baroreceptors start telling the vasomotor system to increase sympathetic nervous drive, then you're going to see an increase in cardiac output, and often you'll see an increase in fluid retention because of that increase in the venous capacitance. Okay, does that make sense? You got more blood pooling in the veins, which allows it more time to cause, swelling, okay? The other things that can happen are dizziness, lassitude, palpitations, and headache, all from the changes in the blood pressure, okay? They can also increase sodium and water retention because your kidneys are going to sense that they're getting less blood flow. So they're going to try to retain water and sodium to increase the blood volume. Okay. They are more attractive when it comes to lipids if you have a patient who has high cholesterol. But they can also cause reflex tachycardia for the same reason I already told you, and increase plasma renin. Don't worry about the renin part here. It's a minor detail. Okay, so this is the drug that I want you to remember, or these drugs I want you to remember, have what is notoriously known as the first dose effect. Okay? About half of patients, when they take the first dose, are going to have orthostatic hypotension. Okay? So you don't want them to take the dose immediately when they get it from the pharmacy. Okay? And they're driving home, or they've got to go back to work or pick up their kids. You want them to take the dose when they're home for a while so that if they get hypotensive, that they don't become symptomatic and have a problem. Okay? They also can distribute into the brain and cause vivid dreams and depression. They can cause sexual dysfunction, which is in men called priapism. You guys heard about that? Okay. It's not a fun one. It is when a man has an erection for more than 4 hours. It gets pretty painful. So they have to go in and get treated for it. Okay. When you give the drugs at high doses or chronic administration, they can lead to sodium retention, also the vertigo, dry mouth, urinary frequency, lethargy, and nasal congestion because, as you remember from that diagram I showed you about the alpha receptors, that's when the drugs are being not specific for the effect that we want. Okay. Okay, so that's the alpha-1 antagonist. And this is just a review slide that maybe is not that helpful as I was thinking about it. But, well, just a review. You have the baroreceptors. We just reviewed this, okay? And then when blood pressure goes down, there's going to be this stimulus that goes to the brain center, okay, vasomotor center. And then ultimately what happens is that's going to increase sympathetic nervous drive. That's all you need to remember from this slide, okay? So what's inside the brain are these alpha-2 receptors that we would like to stimulate, okay? And recall they're right here. So if we were to give an alpha-2 agonist, then we would inhibit norepinephrine release, okay? And we would inhibit that sympathetic nervous system overdrive. Okay. So just recall that when you, you know all the signaling. So this is a presynaptic receptor, okay? All right, so the main alpha-2 agonist I want to talk about is clonidine. I saw it on your top 200 drugs, okay? And we're going to focus mostly on this one. Methyl-dopa is important for particularly one reason, and I'll go through it just shortly. So they're going to stimulate the alpha-1 receptor, which is going to inhibit vasoconstriction, but it can also cause sedation and dry mouth. Okay, so this drug, clonidine, is used for a wide variety of purposes. It's not particularly popular because of its side effects, but it does end up being useful. So you'll see it available in a multitude of different dosage forms. It can come as a pill, as a tablet, it's injectable, or as transdermal, a skin patch, okay? And what it does for our purposes is it's going to reduce peripheral resistance, which then reduces blood pressure. Now, it'll decrease both stroke volume and heart rate and decrease peripheral resistance. So it's going to hit the whole thing because it's working centrally to inhibit sympathetic nervous drive. Okay. It doesn't interfere with the cardiovascular response to exercise. And I put this comment here because that's in contrast to the beta blockers that we already talked about, okay? Renal blood flow is maintained, and there's minimal to no effect on plasma lipids. So when you're comparing which drugs are attractive in which patient groups, these are things that you want to keep in mind. Okay. The other thing is because it's working centrally, there's also a likelihood of increase the parasympathetic nervous system. Now, what do you remember about the parasympathetic nervous system? Anybody remember? Does it work on the periphery? No, it's mostly working at the level of the heart, okay, in the atria. So in that way, it can decrease heart rate, okay, by increasing vagal tone. Okay. You can have an compensatory increase in sodium and water retention because that would decrease blood flow to the kidneys, all right? And in patients who have poor renal function, it would prolong its half-life. So for this drug, has anybody seen it used for anything other than hypertension? Yeah.
I've definitely dispensed it for children before. Isn't it used to help with like ADHD in young children?
Absolutely. It's a common drug for treatment of ADHD. And that's because it decreases sympathetic nervous drive. So about half patients are going to, some of the side effects that you see in the children as well as adults, but about half the patients are going to experience dry mouth, which is called xerostomia, sedation, and mental depression. So not particularly fun drugs to be on. They'll experience bradycardia, okay? And this is another drug that you want to be particularly careful with, having a patient who would suddenly stop taking the drug, okay? Because again, you'd have the upregulation of all the adrenal receptors and blood pressure could very quickly become elevated. So it's used for many other purposes. It can be used for diarrhea, hot flashes, because if you think about hot flashes are another sort of syndrome where people are feeling like this sensation of having increased sympathetic nervous drive, okay? It's used for drug withdrawal. Okay, if you think about the use of illicit drugs, it's not much different than if you're abusing a drug and you overstimulate those receptors through the use of those illicit drugs. Then with withdrawal, they would have those symptoms, and to prevent some of those symptoms from occurring, clonidine can be used so that they don't have that sympathetic overdrive. Okay. Okay. So methyl-dopa is the other alpha-2 antagonist I want to talk a little bit about. Hint here is just look at its name, methyl-dopa. Okay, so dopa is going to be dopamine. Methyl means that there's an added methyl group. So it's going to get metabolized into a drug that looks like norepinephrine. It's going to be methyl norepinephrine. And in that way, it's a false neurotransmitter. So it competes with norepinephrine, but it doesn't activate the receptor, which means that if it occupies the receptor but doesn't activate it, you would get a decrease in cardiac output and vascular resistance. But every time you try to do that, again, this is a pattern here, sodium retention and water retention. So why we can't use it alone. Okay. One of the attractive things about this drug, though, is that it can be used during pregnancy. So I talked to you a little bit about how some women can have elevated blood pressure during pregnancy, especially late in pregnancy. So this is one of the drugs that can be used. Okay. So it's going to decrease sympathetic outflow and thus decrease blood pressure. Okay. And then blood flow is maintained with this drug much better than the other drugs, renal blood flow. So side effects are kind of, yes?
So is the methyl-dopa basically like telling the brain, oh, you've got a lot of sympathetic stimulation going on, let's like down-regulate that? Okay, that's what
It's almost like a blocker, but it's not necessarily a blocker. It's a false neurotransmitter. So does that make sense? Okay. So the other thing, remember with this drug, you guys have had enough neuro, right? So if you think about dopamine has a lot of central nervous effects, right? It's a very important neurotransmitter in the brain. So when you give this drug, it can behave sort of interfere with the dopamine system, so you can end up with Parkinsonian signs. Makes it a very unattractive drug. Hyperprolactinemia, okay? Bradycardia, and then some of these other things like hepatotoxicity. Now this positive Coombs test, this is an allergic reaction to the drug. Okay, it doesn't happen very often, but you have to sort of be aware that it can. So you don't really use this drug chronically. It's one of those drugs that you use more of a short-term use. Okay. All right. Any questions on those? So this is going to move us away from that's all of the drugs that interfere with the adrenergic nervous system. Okay. That's the beta blockers, the alpha-1 receptor blockers, and the alpha-2 agonists. We don't target beta-2. Okay. So now we're going to talk about the drugs that have a direct effect on the periphery, on the blood vessels, okay? And that drug is hydralazine. Okay, so what's attractive about hydralazine is the drugs we've talked about so far seem to affect both the arterioles and the veins, right? So when we affect, what do you think would be most attractive when we're trying to reduce somebody's blood pressure? Arterial blood pressure, right? Because we don't always want to reduce preload, because if we reduce preload, then the heart is going to compensate, increase cardiac output, and we end up with the fluid problems, right? So here, the benefit of this drug is it tends to work more on the arterioles. And how it does so is it works directly on the arterial smooth muscle. It's going to increase cyclic GMP levels, which then is going to decrease the phosphorylation of the muscle myosin light chain, okay? During the congestive heart failure unit, I'm going to talk a little bit more how it has a second mechanism of action, but don't worry about it right now. It's thought to be a nitric oxide donor, and in this way it might also cause arterial vasodilation, okay? So it does not relax the venous smooth muscle. And what I want you to be able to do is to compare and contrast the different classes of agents. Okay. All right, so when we do this, what can happen, the baroreceptors are in the arteries, right? So again, a reduction in blood pressure is going to increase and lead to a compensatory increase in heart rate, plasma renin activity, and fluid retention. Okay, so it's going to dilate the arterioles more than the veins. When you don't dilate the veins, then there's less of a risk for the postural hypotension, like when you shift positions and you get dizzy, which puts these patients at risk for fall, okay? The other issue is that it's metabolized, okay? So it's acetylated in the liver, and about 16% of the 60% of the population are what we call fast acetylators. So they're going to metabolize the drug quickly. And that means the half-life will be shorter, whereas 35% are slow acetylators, and that metabolite is inactive. So that means that the dosage frequency might need to be different in one patient than another. Makes it harder to monitor. Yeah?
The fact that the half-life is so short, because you said only an hour, how does that affect like the day-to-day, realistically, like taking it?
It means that you would be giving it more frequently than probably patients want to. Like, there's multiple reasons why this drug isn't first-line drug. One of them would be you have to take it frequently. You don't know if your patient's a fast acetylator versus a slow acetylator. And then some of these other reasons I'm going to list right here. But one of the things that make it useful is that when we have patients who come in with elevated blood pressure, like, they're symptomatic and they're coming to the emergency room and we need to get their blood pressure down quickly, this is one of the drugs that can reduce blood pressure quickly. Okay? So this is why, even though we have an unattractive drug, it's useful for very specific purposes. Okay? So related to its pharmacologic effect, meaning its reduction in the in blood pressure, patients can have headache, nausea, flushing, palpitations. They can even experience myocardial ischemia when the blood pressure goes down too much. Okay? And then you end up having a reflex in the baroreceptor and increase in the sympathetic nervous system. It too can cause a sort of immune response where it's like drug-induced lupus or serum sickness. Okay, so some patients have antibodies that can react to this drug. And if they don't at the beginning of the therapy, they can develop it over time while they're on the drug. Okay, so increases in the duration or the dose of the drug can increase their risk for this autoimmune effect. The autoimmune effect is also more common in women, in Caucasians, as well as slow acetylators because they have more drugs circulating for a longer period of time. Okay. Okay. So here is a new drug, this dopamine 1 antagonist. I don't think we've talked a lot about dopamine in the periphery during the cardiovascular unit, but there are receptors in the periphery, and we do use dopamine itself during congestive heart failure treatment. But this one is also used as a drug that can reduce blood pressure. And for many reasons, it's a very attractive drug because in this next slide, I'll show you where the dopamine 1 receptor is. Here we have the presynaptic nerve terminal where the norepinephrine is released, and the dopamine 1 receptor is right here in the postjunctional vascular cell. When that is stimulated, it causes vasodilation. Okay? So it would be attractive if we could develop an agonist to this dopamine 1 receptor. And that's what they've done. And it's a very nice drug in many ways because it increases renal blood flow and sodium excretion, which is different from the other drugs that I've talked about. It maintains and improves glomerular filtration rate. It can increase heart rate, and it is not metabolized. It's 90% eliminated in the urine. So what makes this drug not a primary drug for treatment of hypertension is its half-life. It has a half-life of 5 to 10 minutes. So when you have a half-life that short, how do you have to give the drug? Intravenously. Okay, so it's for hospital use only. Again, for one of those urgent situations. Okay, so we've hit all the main classes. I was actually worried that I was not going to get done on time today. I just wanted to go through this one because I'm not sure if you're going to get it later in your curriculum, but it's an older drug. It's a potassium channel agonist, so minoxidil. You may have seen it in the pharmacy. Where? Yeah, okay, so it was a long time ago, it was used for treatment of high blood pressure before we had better drugs, okay? And one of the side effects that a lot of people experienced was increase in hair on their body, okay? So now it's not very commonly used for treatment of hypertension. It's more commonly used as a topical drug for the treatment of balding, okay? And for my purposes, I'm not going to examine you on it, okay? Just wanted to bring that to your attention. All right, so this is sort of a review of the classes of drugs that we've gone over. We've gone over the centrally acting antihypertensives. There's 2 of those. Can people tell me what they are? Huh? Methyldopa and clonidine. Those are your alpha-2 agonists. Okay, this is your quiz sheet here, okay? We've gone over the beta blockers, okay? Things I want you to remember about the beta blockers: 1st generation, 2nd generation, 3rd generation. ISA activity, okay? All right. Beta blockers also work to some degree on the kidneys, okay, down here. Okay, we didn't go over the diuretics, okay? Those will come later. But we did go through the drugs that work on the renin-angiotensin system, right? So in that group, we have the ACE inhibitors, the ARBs, and the renin inhibitors. Okay. Then we went through some drugs that work directly on the arteries, okay, on the peripheral resistance, and I want you to understand those drugs. Hydralazine, okay. Anybody else think of anything else? Any of those drugs? Oh, we missed, yeah, so we did clonidine, right? Okay, so then hydralazine, I can't even remember. Okay. Well, and we had the dopamine 1 agonists, okay, and the alpha 1 antagonists, okay. All right, so that's all of the drugs. Does anybody have any questions? Yeah.
When the picture says that beta blockers reset peripheral resistance, what does that mean?
Which slide are you talking about?
That summary slide that we were on with the brain and the. Yeah.
This slide?
Yes. On the capillary system, that little box says beta blockers reset peripheral resistance, and like, what does that mean?
I don't know. I would ignore that comment. I just want you to mostly, here's the thing. Remember that beta 1 blockers hit the heart primarily. To some degree, there are beta 1 receptors in the kidney. There's 3rd generation beta blockers that do more than just hit the beta 1 receptor, okay? That's all I want you to know. For the non-selective beta blockers, they have indications that, still make them useful, but you wouldn't see them commonly used for the treatment of hypertension. You would see mostly the 2nd generation and 3rd generation drugs, okay? All right. Thank you. Email me if you have any questions.